Hola, buenas tardes. Mi nombre es Jorge Archer. Estamos en esta ocasión iniciando una nueva sesión de Hablando de Huesos. Esta será la sesión número 12. Y en esta ocasión tenemos al doctor Robert Power. Él nos va a dar la plática en inglés. La presentación se tradujo para que pudiera ser más accesible. Hi, Robert. Welcome. Um, I have
Hi,
your
it's great to be able to give this talk. I hope you can see my slides, but I'll try to speak slowly and just to make it kind of easy to follow. But if anyone finds that I'm speaking too fast or it's hard to follow, just you could probably send a message and I can slow down. But we have the slides in Spanish, of course, as well. So hopefully that will help things.
Sure, thanks a lot. If you can talk about your work before, just a bit about what you have done, what's your studies, where do you work right now, and what's your field, please, Robert?
Sure. Well, just by the way, can everyone see my face, or is it just the slides that you see?
Yeah, we can see your.
Right. Okay. Well, so I did my PhD on dental calculus. I'm interested in human evolution, and as an archaeobotanist, I was very keen to learn about diet using ways that we can find information about diet that we don't normally see. So my work in particular has focused on using alternative ways of looking at diet, particularly dental calculus, as it means to track kind of key information about human evolution. So without further ado, I can begin my presentation.
Sure.
By the way, feel free to ask questions at any point. That's fine too. So just as an outline, I'll just give a brief mention about the importance of diet and why, in particular, plants are so important to understanding human diets, particularly the evolution of human diets, and how the study of plants in human diets can be quite neglected due to methodological problems. So I'll then take into account how they're important for studying human evolution, particularly for answering big questions in human evolution, such as the rise and disappearance of Neanderthals about 40,000 years ago when modern humans came into Europe. And then I want to look at some of the other ways we can look at plants and how the role of plants has changed over time, such as plant processing and generally food processing as a whole. As well, I want to look at other applications of dental calculus and how we're developing the methods, as well as trying to use it to understand as broad an array of questions as possible, such as the social role of diet. So in our own diet, and I guess across human cultures, plants are an incredibly important resource and form the basis of most diets and economies since the transition to agriculture. But plants are also very important to hunter-gatherers, and for all sorts of reasons, the role of plants to hunter-gatherers has been somewhat neglected. But in many situations, particularly in warmer environments, plants are a key component of diet, and very often the main foodstuff, with other resources such as fish and meat coming somewhat after them. So the role of plants and the role of plants in early diets has enormous influence in how people perceive food today, and there's kind of a never-ending interest in recreating ancient diets to kind of optimize human health, to seek out some kind of an ancestral diet, this kind of idea that the foods that we evolved to eat might be able to offer us some kind of homeostasis and optimal health. One interesting example of that is a raw food diet, where adherents look at the long time in our evolutionary past where fire wasn't available and where cooking, of course, wouldn't have been available. And they use that to infer that we shouldn't be cooking food, and that cooking food will deny us various micronutrients and maybe macronutrients, and they'll help. Sometimes it's including meat, sometimes it's not, sometimes it's only plants. And although the physiological and health basis behind such diets are questionable, they do raise the question about how important ancient diets are to people even today. Another even more famous example is the paleo diet, which has really gone so mainstream at this stage. And in fact, as far as I understand, the word paleo diet is even copyrighted by certain researchers in the United States, particularly enough. And but this diet tries to kind of strip out the changes to diet that have occurred over the last 10,000 years since the traditional agriculture. So it's really eliminating dairy foods and to some extent removing processed foods and cereals and replacing them with sometimes more meat, sometimes more whole foods, more whole food veg. So that's another kind of interesting example of how people make past diets relevant even today. But when we look at primates, whether it's chimpanzees or bonobos or gorillas or other species, it's the overwhelming abundance of plants that is most striking. Modern primates, of course, have varying diets and diverse diets, but and in some ways they resemble our diets. But what's very striking about their diets is how bulky they are. So although they eat many different foods, including many foods that on a technical basis might seem appealing from the human point of view, if you look at the parts that are being consumed and the lack of food processing, their diets are incredibly bulky, incredibly high in fiber, and they have very little in the way of animal components. Meat might be some way under 5%, like 4% of diet. Well, there's another couple of percent that could be derived from invertebrates, while fish are nearly absent or are absent. Sometimes they do a little bit of bird catching or Columbus monkey catching, but predominantly see this great emphasis on plants. So that stands in stark contrast to how people have perceived ancient hominid diets. And of course, we're not descended from chimpanzees or bonobos directly, but we use these species as kind of a crude proxy.
Robert.
Sure.
Sorry, Robert. We have your audio is really bad.
Oh, sure. Yeah. Let me see.
Can you maybe we can stop your camera and we can just leave the slides? It would be easier.
Yeah, absolutely. Yeah. I'm not. So that's on the.
No, yeah, the slides are on. So yeah.
So stop camera.
So,
okay.
Yeah. So I would put just share your slides again. We can leave the
Right.
audio.
Okay.
Slides are on, and I think it's better right now. Thank you.
Good. I don't think you lose too much without my face.
No, actually, it's better. Yeah, sounds better.
Right. Good stuff.
Thanks.
I mean, I can also go a bit closer to my router as well. That could potentially help things.
That's perfect.
Just let me know if there's any problem.
Yeah.
So there are some enormously significant trends in skeletal record of in hominid evolution. For example, the rise of bipedalism, which of course is linked to how early hominins were moving across the landscape and moving to procure resources in more and more open environments rather than being more kind of tree-based species. But there's also this slow incremental change in dentition with a very significant reduction in dentition occurring at various different points. And it's very clear that this change is linked to increasingly, I guess we'd say, energy dense diet that required less processing costs from the point of digestion. And of course, people have looked at meat as a candidate because meat is a very unusual food, and though it varies quite a lot depending on what kind of meat we're looking at, whether it's liver or brain or muscle tissue. But generally speaking, meats are a fairly balanced food that has quite a high nutritional completeness. And there are not many nutrients that occur in meat that are absent in plants, very few, if any. But what's important to stress is in plants, things tend to be more scattered, so we need to consume a broader array of plants, which would require more in the way of foraging costs, location costs, harvesting costs. So people have pointed to meat and as a key dietary change, and this might tie into the advent of stone technology sometime after 3.4 million years ago, which could necessitate new ways of processing resources, because when you, of course, eating meat, you might need to cut the meat, butcher the meat, whether it's scavenged or hunted. So meat has this incredible emphasis in archaeological and paleontological communities, and I guess very simplistically, people have just looked to hunting and meat as this kind of key trend over the last several million years that enabled reduction in gut size, enabled brain expansion, reduction in dentition, and all these different factors. But of course, and this is a very simplistic perspective, and even though meat's a very important food across foraging populations, plants are a very important food too, and plants are enormously important in all different ways. And it's very easy to neglect the fact that plants are often the dominant food in foraging populations, particularly in kind of more warm weather environments that are so key to the kind of evolutionary stages in human origins that we're talking about. The situation in the far north or the kind of extreme south, it's not necessarily as relevant for this kind of earlier time period, of course. And I guess often forages from very cold environments are kind of the more, more historically marginal communities that just appear to be typical today, but not necessarily typical in the past, because they're simply the only foraging communities that haven't been replaced by farming populations. So if we actually survey the diets of foragers and compare them latitudinally, we can see this incredible variation in diet, and also in different types of resources being used and how they're being used, with this very strong trend of increasing dietary breadth in more warm, more tropical environments. So that raises the question: how do we study diet, and what can we do to overcome this research bias that has plagued the field for so long? So, one promising method, of course, is through modeling approaches, using recent hunter-gatherers to develop kind of a theoretical model. Of course, this approach is very environmentally deterministic, and it assumes that recent foragers are necessarily representative of the past, and that is simply not the case. There's all sorts of technological innovations that occur in different regions at different times that will shift the kind of resources that people are consuming. For example, technology such as fishing boats, fishing hooks, were not available several million years ago, but their advent allows much greater use of the fish. So it's very obvious that a modeling only approach is far too simplistic, and sometimes we need to find direct evidence of diet. And the further back we go in time, the more difficult it is to use traditional archaeobotanical methods, because very often there's simply no archaeological site to apply methods like archaeological flotation. Habitation sites are so small and ephemeral that the only evidence that we see is very, very thin palimpsests, such as lithic layers in cave sites that often isn't even primarily deposited there in the first place. And these sites are subject to all sorts of carnivore activity and disruption, as well as all sorts of destruction during glaciation periods. So one of the ways we can get around that is to use microbotanical remains, particularly microbotanical remains that are highly robust. And one of the most principal ones that I focus particularly on are phytoliths. So phytoliths are just one of the proxies that we can use. There's also starch, which of course is the very energy storage organ of plants themselves. It's particularly abundant in roots, but you can also find it in leaves and elsewhere. And there's pollen. And pollen is highly durable. It's excellent in many ways. You can find it in all sorts of environments. The problem with pollen in this context is that it's reflecting the sexual organs of the plant, and it's not necessarily the resources of the plant that were important from the point of view of the forager. Because the other issue with pollen is that it can be designed to be dispersed very long distances through wind or through insects or other dispersal agents. So it's very prone to traveling long distance and kind of presenting kind of a misleading signal from the point of view of diet. Although it's ideal from the point of view of environmental reconstruction, it's a bit different when we're looking at diet. So that leaves us really with phytoliths as a very robust, low contamination risk way of looking at diet. The only trick is where can we find them? Where can we have a stable environment that we can actually use to reconstruct diet from? And of course, that's where dental calculus comes in. and why it's such an exciting material. Dental calculus is mineralized dental plaque. It's dental pathology, but it's also ubiquitous, and it's often very well preserved on archaeological sites. The only situations where we don't find it so well are situations where the dentition is not so well preserved, or where there's been a lot of alteration of the dentition, or perhaps where there was not much calculus in the first place. So studies of dental calculus have showed that it's a very, very stable environment, and it's very robust to decay and alteration. And the layers, there are layers of minerals that can preserve molecular and botanical data encapsulated inside that is highly resistant to alteration, unlike the archaeological sediments that you might get at an archaeological site, or for example, somewhere like maybe a stone tool. They're very popular places like for phytoliths and statues, so they're very prone to alteration because they're such an open environment. And we think that calculus is a closed environment, and therefore that it's much more secure. And when we're dealing with something so small as a phytolith, knowing when an environment is secure and that's not open is really key to being sure there's no contamination, because even a little contamination can skew results quite far. Dental calculus forms in all teeth, but it tends to be particularly prominent on the canines, or in some of those premolars, such as in this case, because that's where we have the salivary glands located on the roof of the tongue. So actually, dental calculus has been noticed for a long time by archaeological researchers, but it hasn't really been taken very seriously for so long. Early research looked at dental calculus from the point of view as a sign of ill health, kind of a general indicator of poor oral hygiene, and we still do that today, but less so because the information that I can tell you about the health is quite unspecific. And although it's, I guess, can be seen as dietary driven, it's quite unspecifically dietary driven. For example, both meat and hide and plant use are associated with increased dental calculus formation. So it's a very unspecific pathology. Some of the early uses included work in Southwest United States in the 1920s. There's kind of been continuing use since then, and since about the 90s, there's been a lot of interest in dental calculus as this kind of secure depositional environment. So we now understand that dental calculus is a very, very complicated matrix that's chock-a blocks with all sorts of information reflecting the life of the individual. Predominantly, it is bacteria, but also we have DNA of the host, also human proteins, food proteins, viruses, and food, as well as, of course, food DNA too, and phytoliths and all these other factors. If we think of calculus, or if we think of teeth generally, what's really interesting about teeth is that the only surface, I guess they're the only interface we have with the environment that's non-shedding. So it's kind of a unique situation from childhood till death, that they kind of capture all the kind of series of interactions. So of course my presentation is on calculus, but of course there's just more generally, I mean, there's just a whole ream of information that we can just learn from tooth surfaces. So kind of that takes us to Neanderthals, and Neanderthals, of course, have a very long, complex history emerging from populations that moved to Eurasia from Africa at a unknown point before evolving into Homo neanderthalensis. And we know Neanderthals had a very complicated population history of kind of ebb and flow, expansion and contraction. But overall, they occupied Eurasia for a very long time, and they had an extraordinary range from Portugal in the west to essentially the Russian border with China in the east to the Sinai Desert region of Arabia. So we often think of Neanderthals as being an extinct species that disappeared because of modern human arrival in Eurasia. But Neanderthals, of course, were very successful in their own way for a very, very long time. And I guess the question that I've been interested in is, how did Neanderthals last so long in Eurasia, coping with all sorts of periods of climatic and other stresses, including dramatic swings in climate, then only to quite suddenly disappear when modern humans arrived. We know now that to some extent Neanderthals survive as a mixture in human populations, but by and large they quite abruptly disappeared when modern humans entered Europe and the Near East. So there's been a lot of ideas that Neanderthals were somewhat an unsuccessful species, or a species that were kind of doomed to disappear because they had a very narrow or constricted diet that was maybe too kind of top heavy. We know that they were very specialized though in some respects, because they, across their range, they seem to have exploited small and medium sized ungulates, but they were also able to hunt the biggest, many of the biggest species that occur in the environments that they occupy, including mammoths. So we also know some information about their the other aspects of the diet. There's some evidence that they use shellfish, but there isn't systematic evidence of it, and it's not clear if this is because of rising sea levels that it might have hidden the evidence. But generally, people think there's not a lot of that happening, particularly based on isotopic evidence. But we also have some information of plant use, particularly in the southern range, such as Ahmad Caves in Israel, in Gorham's Kale in southern Spain, Gibraltar area. And these indicate something about the flexibility of Neanderthals in warmer environments. And so I was very interested in to see, does that play out in the same way when we look at more data, more methodologies that can explore plant use. So that's where dental calculus can offer a very special opportunity. So just kind of looking more in depth at the upper Paleolithic diets as contrast, there's been quite a lot of isotopic work, particularly from the kind of Gravettian period and later, and this shows that upper Paleolithic people were also very, very successful hunters, like Neanderthals, but they also used plants on some occasions. In some cases, we have evidence that they're using a lot of fish-based foods from freshwater and sea environments, but generally they seem to be quite terrestrial. So my own work has looked at upper Paleolithic diets too, using calculus to kind of get a perspective, or like I guess a comparison point. And we can look at sites such as Gravettian era occupations of Stalin, Vestinici, and Pavlov, and we can see that even in these very cold environments where these Gravettian hunters were seem to be really specializing in the exploitation of steppe grazers that were occupying the steppe. They were still using some plants, and they seem to be using plants for a variety of purposes, including maybe fiber or kind of textile productions, so not just food. So we also can see evidence of paleolithic diets during warmer periods. For example, at El Meron, and we can see that during these kind of warmer periods that are really approaching the end of what we call the Upper Paleolithic, there's really fluorescence of kind of plant specialization and a much broader range of plants being used. And of course, you also see specialization in terms of the hunting economy, such as the very strong evidence of ibex hunting. So to kind of look at this from a theoretical point of view, I wanted to compare Neanderthal diets to different, I guess latitudinally, but I guess the trouble is that Neanderthals occupied Europe during different climate oscillations. So to take into account that climate change altered each occupation substantially at different points, we assigned a biologically relevant measure of temperature called effective temperature to each occupation and each archaeological specimen of Neanderthal, where I could get dendrocalculus. And I used this method on recent hunter-gatherers just as a kind of a test case, and I was able to show that this method works for hunter-gatherers. You can see that in warm environments, measured by effective temperature, you see a broader diet. So with that kind of cleared, I was able to apply that to dendrocalculus from a variety of sites, particularly some very important Neanderthal sites such as Vinja and Scutari and Fasoloni and Sena de los Huesos. And these sites are important because they help kind of look at Neanderthal occupations that were in some kind of intermediate environment. So we have evidence of plant use from the warmest environments sometimes, based on the macro botanical remains, but from the isotopic evidence, there's just nothing, because it doesn't, because isotopes, it's not possible in these kind of very early warm weather sites, and also because people haven't tried. And also these sites kind of indicate they offer a perspective into more cooler environments, which this dendrocalculus studies have also contributed to, such as Kolna and Farsi, amongst other sites. So using dendrocalculus analysis, we're able to retrieve a variety of microbotanical information showing evidence of seed use, different nuts, things like lily root, as well as birch pollen, different endosperm seeds as well as pollen from ferns and also cereals. So I guess the challenge with microbotanical remains often is that you can have some microremains that are not easy to identify, or they're not necessarily distinct, and you might have several microremains from one plant, and then another plant might have no microremains. So to kind of take that into account and to look at this from a statistical perspective, because most people doing this kind of work aren't applying much statistics, we developed kind of an index, which I called minimum botanical units. So just like a minimum number of individuals in human osteology, and that allowed me to kind of get a more precise estimate of dietary diversity in each site, well, per an individual on each site. So sometimes you'd have several measures on each site. And this allows me to kind of chart out, I guess, dietary diversity across our range over different time periods and during different climatic phases. And it showed, although it's not very clear from here, but using a linear model, we were able to show that there's a fairly stable kind of Neanderthal niche across their occupation. And it was used to infer that even in cooler environments, they were using plants just like they were using plants in warmer environments, and that you have to be careful about assuming that Neanderthals were not necessarily narrower diet than the other Paleolithic populations that replaced them. It also showed that the way that we think of plants can be very simplistic because it's based strongly on ethnographic analogy, and that it's what's very important is to consider how the plants are used, and that's something that may or may not be possible for microbotanical assemblages. So we found foods that, for example, grass seed, that are more associated with much later populations. But I think when you take into account that Neanderthals may have had their own ways of processing these foods that were less intensive and that were more high return in their cost to benefit ratio, they would have they would have made more sense from a Neanderthal energetic model. So the interesting thing about this work that has kind of continued since the publication of the work and the completion of my PhD is that I've continued this work and I've continued to look at kind of the less studied environment. So fairly recently, it was realized that Neanderthals have a much broader range than people once thought. So we know they also occupied Central Asia. And we don't know a lot about their occupational history in this region, but we think that they favored kind of transitional forest steppe and steppe environments kind of along mountainous regions that may have been similar to their Western Eurasian range. So quite remarkably, we found kind of that even in this area, even in the middle of Central Asia, on the border with China and quite near to Mongolia, that they were still using a kind of a hominin dietary niche that was reminiscent of Western Eurasia. And we had this pattern also play out in the isotopes and the zooarchaeological remains too. So the challenge with doing this research has been that with depending on fossil evidence, and actually, although Neanderthals there's not so many Neanderthal fossils available, there's even less fossils for the very earliest people, very earliest modern humans that arrived in Eurasia. So when we take into account the earliest history from about 46,000 years to about 35,000 years before present, there's almost no evidence in the skeletal record apart from a few sites like Oasi. So one of my kind of passions has been trying to look at those very seldom found sites where we can get evidence of the kind of earliest modern humans, what we call the initial of a Paleolithic or later on the Orignation, and apply isotopic, denticalculus, and other methods to infer their diet. The problem is they're so seldom available, but my work has also applied these approaches to the site of Platica cave in Bulgaria. And the site is like the earliest site where we have clear evidence of modern humans. So it's able to show that perhaps in these very early occupations, our perspective of what a Paleolithic person's diet may have been isn't necessarily true, and that they may have been initially kind of more like the kind of the stereotype that we have of a Neanderthal diet compared to the later populations that we see, for example, the Gravettian period or later on again. And it's very likely that these early initial populations were so low density, so sparse on the landscape, that they may not have had the need to develop kind of economic specialization, such as fishing, such as specialized hunting methods, for example, or more intensive resource strategies involving plants. So a lot of my research has focused on using, I guess, different plants to understand advents of new behaviors in the hominid record. But I really wanted to just put a few slides in this talk today about how it's not just what we eat that's important, but it's how we eat it. So, a calorie is not simply a calorie, or a kilojoule is not simply a kilojoule. This sample, a slide sample, just shows how nutrients can be consumed, but they're not necessarily exploited by the digestive tract. In this case, it's almond cells from an uncooked almond, and we see in the uncooked almond the nutrients are still trapped in the robust plant cells, despite being consumed and passed. So it's very important to consider how foods are used. So there's also, we have to take into account our microbiomes, and our microbiomes have direct input in the amount of nutrition we're getting from our foods, and something that we can't recreate. So it's an important caveat. But we can recreate some aspects of how people ate food in the past. We can look at robust technology such as groundstone technology and compare it over different periods of time and see how it waxes and wanes, or increases and contracts depending on different factors. And what's interesting is that we see a similar pattern compared to what I mentioned earlier, where the very earliest sites don't necessarily have a very clear change in how groundstone tool technology is used. So of course, groundstone technology is heavy and mobile, but it offers advantages in that you can process nuts and seeds and make them more nutritious, make them more energy dense. Of course, there's a cost, but it's generally seen as part of a suite of resource intensification strategies. So I think it'd be just nice to look at something completely different, just to show some more of my recent work. And I just wanted to look at how we can also use dental calculus to study diets in warm weather environments. I'm very interested in prehistoric dispersals of food that are unexplained. So one of the really interesting examples is the prehistoric dispersal of banana, taro, and yam, sugarcane, which are Southeast Asian domesticates. And these crops appeared, of course, in Africa in prehistory, and they've utterly transformed life in the African tropics, just like they've transformed life in the neotropics in Latin America. And this is a very good case of application of microbotanical remains, because a lot of these foods are quite rich in phytolates and starches. So one of my most recent projects has been taking into account what we can see from dental calculus, but also C4 modeling, linguistics, and genetics, and trying to use that to kind of reconstruct how this crop dispersal occurred. So as part of this project, I've looked at dental calculus from a lot of sites in Central Africa and East Africa, and we've gone to find evidence of banana from a site called Nadali on the Nadali Lake area in central Uganda. And what's really interesting about the site is that we looked at many sites for evidence of banana from early sites, and we expected to find a lot of evidence, but just none is showing up. And a lot of other people have looked too. There's been looked at all sorts of different methods, such as sediments, also calculus tools, and no evidence is showing up, even though people have thought that that banana appeared very early in prehistory. But this work with dental calculus is showing that there's probably a much later dispersal that then realized, but it's a dispersal that hasn't been linked to population dispersals or crop exchanges across the Indian Ocean in a kind of concrete way. So there's a lot to figure out, like how did it get to Uganda? Did it come along the Swahili coast? Was it through Ethiopia? Was it through India, which was an early user of banana, as well as the other Southeast Asian foods? So I'm also interested in developing dental calculus for more broader approaches. So one of the issues we have is that the research is fairly destructive or semi-destructive, and dental calculus is a very unique substance with a lot of information about the past. So we want to be as undestructive as possible, and we want to look at or like see inside the calculus without damaging it. So we're developing ways which we can pick out information from the dental calculus using a micro-CT approach, and using that to kind of have a non-destructive approach to dental calculus analysis. So almost finished, but just lastly, I just wanted to mention diet and social roles. So I think with dental calculus, it has a very unique perspective in that we're looking at the diet of an individual, and with an individual we can look at how individuals lived, and we can do things like osteobiographies, reconstructing the life of how people removed from the generalities of a group. And one of the kind of interesting ways which that can play out, or that can be exploited, is by trying to look at gender differences in diet and health. And using dental calculus, if you have a big enough sample size, you can begin to get at questions like diet and gender, and like what role of diet over life history. Did younger individuals have different diets? Can we detect weaning? And can we detect dietary transitions as at different stages in life history? So anyway, that's my presentation. I just wanted to give a kind of brief overview with a kind of a main focus on Paleolithic diets, but there's much more than the Paleolithic. So like I know a lot of the people following the group would be interested in more Holocene history, so I wanted to just give an insight into how we can provide information even for quite recent periods. So I just have a brief thank you slide, and of course thanks to all the people who helped my research, but also to the host for kindly inviting me and allowing me to disseminate my work in this lovely way. So I'm delighted to hear questions, or maybe there's things that were hard to follow because the sound quality wasn't as good. And if you want to ask about any individual points on any slides, feel free to go ahead or to ask away. I can turn back on my camera now as well, so it's just a bit
Yeah,
easier.
you can put it back. Thanks, Robert. Okay, thank you so
Okay,
much.
so hopefully
So yeah, it sounds very kind of has some cuts on the transmission, but well, it's understandable. Either we're giving up maybe the next week a small chat about the same theme but applied to Mesoamerica because there's a lot of questions about Mesoamerica. And bueno, si alguien quiere hacer preguntas, con todo gusto las podemos contestar, ya sea en español o en inglés, como ustedes gusten, por favor. Okay, so the question is going to be displayed on the screen. If you want to read it, or I can read it for you if you want. Robert?
Okay. Yeah, so in some cases we can definitely find evidence of that. So, for example, in one case there was a study where they found a lot of evidence of palms, and they knew that palms were no longer in the local environment. They were previously there, but they had been wiped out through deforestation. So they were able to say that there's definitely palms getting in through sediment that was not relating to diet. I think it depends on the case by case, but I think generally speaking, water probably isn't a major contaminant because water wouldn't have so many micromains. Now, sometimes that's not the case, but I think generally speaking, activities like basketry are kind of a bigger source because with basketry, you're working with materials that are actually usually rich in phylates, and you're kind of manipulating them in your mouth. So there's a very real opportunity for things to get embedded there. But there's various ways you can take that into account, and I think the most important thing is to once you identify what you find and you have kind of you have it present in multiple samples, then you have a chance of picking that out and taking precautions to avoid confusion.
Yeah, well, there's I think we were discussing last year about the possible contamination that we had, for example, with the fireplaces and all these, that you breathe the smoke and you breathe the small particles of charcoal or carbon, and they get stuck in between the teeth. So that could be also something that could be changing some of the information, but still it's a very valuable information that you can get in the research.
Yeah, and smoke is a big one. Definitely smoke has a massive impact. Because sometimes you can use mass spectrometry to identify smoke, but I think smoke could probably be putting in graphite lit sometimes in some areas as well.
Yeah. Okay, so we have some people that was connected. Erich Sorn, he said that just it's in Spanish, but it was it says that including in Mesoamerican archaeology, most of the vegetable remains are forbidden. So it's quite common. It's something that is been coming back in these times.
What do you mean by forbidden?
Yeah, that sometimes people just don't look at them. They were just like in the past, or there's no one studying them, or right now they're getting more interest, obviously.
Good to hear.
Okay, we have. Okay, this is Francisco Antonio Sorio, and he says, "Thank you for the information. It results interesting that here in Mexico we know from a lot of time ago the potential of the analysis of the endocalculus, but in this presentation it's more visible that the studies have a regional effect or effect that from the ecological point of view, presents more potential for the human adaptation studies. It's just a comment.
Yeah, so I mean, I guess it's case by case. I mean, I think phylates have been important for tracing the history and kind of emergence of Mesoamerican crops. That they haven't been used for that purpose with dentocalculus so much, but I think it could potentially be useful. There's certainly tropical crops in Asia that I think could be traced. Like their domestication could be traced for calculus, and yeah, I mean, it's I guess it's case by case really, because you know, sometimes the wild progenitors are very common forage foods, sometimes they're not. If there's a big difference between the two, like wild banana isn't really a common food for hunter-gatherers. It's just not used that much. So if that's the case, then you know you have a fairly clear-cut case where you can use it to trace adaptation and maybe domestication or cultivation.
Yeah, that's correct. Okay, there's another question. Gabby Batch. Hey, everyone, could you explain a little more about the micro CT methodology? Thanks, Robert. Nice talk.
Oh, well, it's very experimental. I mean, micro CT has a lot of potential. The trouble, like the difficulty, is that like a lot of CT doesn't have the sufficient resolution. So we're trying to use phase contrast, and it's, to be honest, it's not like a mature methodology. It needs time to be refined, but at the moment it's a fairly labor-intensive technique, and using kind of traditional methodology might be easier for most studies. Maybe in the future it'll get more cheaper, but I guess it depends on the institution. But certainly in Germany, it's quite expensive.
Well, it has something to do with it. Guillermino Ortega says, what's the kind of methodology you use for the study of dental calculus in prehistoric remains? What's the methodology you use for studying dental calculus on prehistoric remains?
I completely skipped over that. So it's possible to look at a dental calculus using a microscope, particularly a SEM microscope, and we can sometimes identify microremains on the surface. That's a really great method when it's something that you don't want to damage or want to have minimal intervention. But generally speaking, we sample the dental calculus and we decalcify it, and we try to leave some on the dental calculus on the tooth, and just take a little bit, and then we can really go through that matrix and break it up and see what's there. But I mean, there are different ways to break up the matrix, and actually each method has its own advantage and disadvantage. So it's something that has to be really tailored to the question you want to do.
Ya, bueno, si quieres lo contesto rápido en español. En este caso, bueno, obviamente hay varias técnicas. La más usual es la de, pues se puede tomar, bueno, una no tan usual sería la de tomar una muestra y ponerla en un microscopio electrónico de barrido, y se puede ver alta resolución lo que quedan de restos en el cálculo dental. La otra técnica que es destructiva, se puede tomar la muestra de cálculo dental y en este caso se puede diluir o descalcificar por medio de varios métodos. En este caso se puede usar, por ejemplo, EDTA, que es un compuesto que se usa principalmente en genética, o bien se puede usar ácido sulfúrico a muy pequeñas concentraciones, y esto a un tiempo prolongado desmineraliza y después puede ser observado en un microscopio óptico común y corriente, y después ya entraría la identificación de cada tipo de fitolito para poder generar bases de datos y esto poder interpretarlo posteriormente. ¿Alguna pregunta más? Okay, well, I think there's no more questions.
Okay.
Well, that technique that you were telling about the micro CT scan is very useful. It's quite new. There's another one. It's El Chagoya says at the conference. There is an option that the carnivorous resource of the upper Paleolithic comes from marine resources.
Yeah, in some cases, yeah. Like the best way to look at that is, well, the traditional way to look at that is stable isotopes, and in some situations that works well and we can detect it. Now, since the end of the last ice age, there's been sea level rise, so the populations that were probably using the sea the most are underwater, and so that makes it quite hard. Unless you're looking at somewhere that hasn't been much sea level change, such as some parts of like southern Spain or Greece. It's likely that there was much more seafood use by Neanderthal than we expect. That's kind of hidden by this factor too.
Okay, thank you so much. Okay. Ok, ¿alguna otra pregunta? Con todo gusto la pasamos. O no puedo quitar esto. Ok, perfecto. There's a delay for about 10 seconds with the Facebook page, so sometimes it takes a little bit more. But, yeah, I was telling you the CT scan technique, it looks very interesting, maybe very promising for the future. There's another one here, and let me put it on display.
Yeah, so...
Okay, is there a proposal for a unified protocol for microremnant extraction, for example, the protein or DNA?
Yes, for DNA. Not really. Okay, so there are protocols, yes, but then they're probably not the best protocol. For proteins, for DNA, yes, it's fine. For proteins, I think the best protocol it has to be done separately.
Yeah, if there's any protocol for in general.
Yeah, so I mean, I work with a group that does a lot of protein work, and until recently they were using a method that that would allow you to extract the phytates probably undamaged. Now that method would destroy the starches, so it's not ideal. But generally speaking, if people who work in proteins or DNA, they want maybe like 10, 20 milligrams, so they want big chunks. And those individuals, particularly in I think in more recent Mesoamerican samples, you can sample something separate for microbotanical remains. So I mean, if you're doing this kind of work, basically maybe it's possible, but it'd be better just to keep a separate sample for each methodology. It's less of a problem with DNA though, but the others it's an issue. were you able to? Okay.
Yeah, I think there's a lack of, well, there's not a, in general, creo que, bueno, voy a poner en español mejor. Creo que en general lo que pregunta Vicky es que si hay un protocolo establecido para análisis de microrestos óseos o, bueno, microrestos cálculo o eventos de extracción. En este caso todavía no está bien establecido, a pesar de que sí se han hecho metodologías, sobre todo en publicaciones de artículos, pero todavía está por explorarse, ¿no? Entonces sí, todavía está desarrollando, hace falta tal vez crear un protocolo internacional para poder hacer la extracción, pero como también es algo que está desarrollándose constantemente y está pasando, cambiando mucho, el problema es que sí, no hay todavía un protocolo como tal. There's another question. I have some issues with my desk. So Guillermina Ortega says if there's what's the dental piece that where you have found the most or the largest amount of dental calculus?
Like the individual or the tooth? Like which type of tooth is it, or like molar or canine? Yeah, probably canine. Canines. Maybe the incisors near the canines. I mean, I think like an M1 is better than an M3, right? Because it's older. But it's not too big a factor in my opinion. But.
Yeah, the distribution changes depending on the diet. It could be just a small sample, puede ser una or a large sample, so it doesn't really matter. Okay, there's another question for you.
Okay. Yeah. So I've been doing
Robert, congratulations. It's very interesting the work you are. Yeah.
So there's been two kind of contemporary studies, one on forages from Namibia, done by my colleague Chelsea Leonard, and also using chimpanzees as well. And we've been able to kind of piece a lot together using these two groups. But it'd be great to get more recent ones too. But there's quite a lot of issues studying populations that are contemporary. Like you have to, I mean, there's a certain amount of legal work to ensure that it's okay with ethics boards. So until now, people have been focusing on archaeological populations, but hopefully there'll be more contemporary populations.
Okay, he has another question. It's very interesting work you're developing. Have you? That's the same. All right. Okay, I think there's. Creo que no hay más preguntas. Nada más hago un comentario, Angie. En poblaciones modernas, sobre cálculo dental, no solamente como pilotos, por ejemplo, la doctora con sobre lo que es el agave, y se toman algunas muestras de una persona que de hecho tomaba pulque, entonces para hacer la comparación con la muestra prehispánica. Entonces nada más, pero no hay demasiados trabajos sobre eso, solamente en cuestiones de citología o cuestiones de desarrollo de sarro, todo esto, pero para cuestiones de fitolitos no hay tanto. Okay. Well, I think there's no more questions, and thank you. Oh, there's another one. Sorry. Let's put this on. Vamos a esta la última pregunta.
Well, I mean, maybe with micro CT, but like, I think most methods of scanning dentition wouldn't be good enough resolution, but perhaps a micro CT is first, but you have to get the resolution correct, like, and you need phase contrasting, so it's quite challenging to do that, but But to be honest, I don't know if anyone has tried it with molds or anything. It'd be interesting to see if they can actually capture information. Like possibly they're precise enough to capture the phytoliths and starches that are stuck to the edge of the cálculo.
Sí, bueno, efectivamente como es muy pequeño la pieza dental, a lo mucho que se puede hacer es tal vez hacer un escaneado CT scan y tratar de hacer la impresión tridimensional, pero la cuestión del cálculo dental como es tan pequeño se pierde, ¿no? Es lo que trata de decir. Bueno, entonces, thank you so much for the talk, Robert. I shared your academia.edu page, so if someone else is interested in looking at your research or the research you have been doing in the last years, they can look at there, and they can see your papers. And, well, thank you so much.
That's brilliant. Thanks so much for inviting me.
Well, we can invite you in the future, and we can just... You're welcome. And, well, thank you for accepting. And don't disconnect. I was going to end the transmission. Bueno, muchas gracias. Entonces, la siguiente semana vamos a hacer una plática igual de este mismo tema, pero en español, para la gente que tenga igual alguna duda o algo así. Con todo gusto lo tratamos de explicar. Bueno, pues muchas gracias. Que tengan buena tarde.